guidelines are being developed for methodology, research, and development of drugs from traditional systems of medicine so that it can act as an alternative or complementary source for medications for intractable diseases. And hence, there is attempt to facilitate and promote the understanding of these systems of health and disease all over the country and all over the world. Medicinal plants are a very important component of traditional medicinal remedies. Now, medicinal plants have since long been source of very important drugs for modern systems of medicine, and a lot of chronic diseases have been treated by products that have sources in medicinal plants. Other than that, medicinal plants used in traditional systems of medicine have also been used for several diseases simply because of the fact that it is cost effective and it has fewer adverse effects. And the complementary role of such traditional medicines to modern medicines are being proposed to fill up gaps in respective gray areas. Interactions of traditional and modern medicine is another very important emerging concept. How to integrate traditional and modern medicinal concepts. So one of the ways to do it is to validate the effects of traditional therapies, which can promote new drug development by preclinical as well as clinical research. The common approaches adopted in traditional medicinal research are A, translational research, which would mean laboratory findings are translated to the clinic. B, reverse pharmacology approach, which would mean that observations seen in the clinic in traditional systems of medicine are taken back to the lab and tested in disease models in order to see as to how these drugs act as they are proposed. And C is a method which is known as repurposing. That is, some of the traditional medicines which are used for particular diseases, listed for being used in particular diseases, are now being used for other diseases because of the basic properties that they possess. So several interventions in traditional systems of medicine are being explained by modern biological mechanisms. Cellular and molecular biology and systems biology approach are being incorporated into traditional medicinal system research. Now, the topic of today's lecture, the example that I've given is for bronchial asthma. Because the respiratory disorders are so common all over the world because of their morbidity and mortality. And bronchial asthma is a disease which involves the airways, inflammation, there is hyperresponsiveness, there is airflow restriction. Current therapeutic strategies have safety limitations, and hence alternative approaches are being explored. Traditional medicinal systems could provide alternative or adjunct therapeutic modes because not only they are cost effective, but also they have fewer adverse effects. Asthma treatment is basically based on corticosteroids and bronchodilators, and they have to be used for longer periods of time, as a result of which we know that adverse drug reactions due to drugs, as well as treatment costs, are a major reason why compliance to therapy is affected. And thus, therapy is interfered or therapy is stopped, and so the disease comes back. You get all kinds of complicated bronchial asthma, which increases morbidity and mortality. Alternative modes of treatment like medicinal plant-based treatments or herbal drugs are hence being explored. Such agents, that is medicinal plant-based agents, have been effectively used in traditional systems of medicine, even in some cases of bronchial asthma, and may have effects per se, or may even interact with the conventionally used modern medicinal agents to reduce their adverse effects or cost. Ashwagandha Withania somnifera is a medicinal plant which is known worldwide. In India, it is known as Indian ginseng. In other parts of the world, it is known by different names. It has been used for 4,000 years in Ayurveda. It has several properties which fit very well with asthma, like adaptogenic, immunomodulator, anti-inflammatory. So all of these pathologies are associated with asthma. But there has been no concerted study to evaluate the effects of Withania somnifera with reference to these properties and their effects in bronchial asthma. Withania somnifera, different parts of the plant are effective. The roots are effective, leaves are effective. Irrespective of the part of the plant, it is the glycowithanolides which are the active components, and they contribute to the biological effects of the drug. Hence, we did some preclinical studies with the standardized extract of Withania somnifera on experimental models of bronchial asthma. So this is your schematic representation of Withania somnifera. You can see that it is anti-inflammatory, it is adaptogenic, it is several other properties are known. So it is the anti-inflammatory, immunomodulatory, and anti-stress effects that we will be targeting when we look at the potential of Withania somnifera in bronchial asthma. So what we did was we studied the effects of Withania somnifera in experimentally induced asthma models. So first of all, we studied it on bronchial hyperresponsiveness, studied its effects on airflow restriction, studied its effects on airway inflammation, on airway remodeling, and finally mast cell degranulation. So this is the experimental design. We used the rat model for asthma. It is a becoming increasingly popular model because of its easy maneuverability, easy availability, and reproducible results. So it is a model of allergic asthma. We have sensitized with the animal with ovalbumin and aluminum hydroxide, and then challenged 14 days later with ovalbumin, and then after 24 hours, it is challenged with inhaled methacholine to induce bronchoconstriction in this particular model. And this, after the model of asthma is established, only then we look at the biochemical parameters that we are looking for with reference to anti-asthmatic effects. So first of all, we wanted to establish the model of asthma. So airflow restriction is the first thing. It was done by whole body plethysmography, where the airflow through the bronchial tract was assessed by a machine, computerized machine, which is known as whole body plethysmograph. And here we recorded airflow restriction, and you can see here that airflow restriction is recorded as P enhance. And in the disease control model, that is immunized and challenged model, there is an increase in airflow restriction. So that means there is bronchoconstriction. And when the standard drug prednisolone is given, that is the steroid, you can see the airflow restriction is reversed. So once we establish the model for asthma, we will go to the next slide. You can see here that this shows different concentrations of methacholine challenge, and on the y-axis you can see the P enhance, which is the enhanced pause, which is an indicator of airflow restriction. And you can see here in the disease model, the airflow restriction is maximum in red. And with dexamethasone and with Withania somnifera, the airflow restriction is reduced. And so this shows you that the model for asthma has been established. When we looked at IgE, which is again a very important immunological marker for bronchial asthma in the bowel fluid, we showed that IgE levels were markedly raised, almost 100 times in the disease control model, and reversed by prednisolone. Similarly, the inflammatory cytokine TNF-alpha levels were 3 times higher in the disease control, which were reversed by prednisolone. And eosinophil count, another biomarker of allergic inflammation, were again markedly higher as compared to controls, and this was also reversed. So all these parameters, basic parameters, showed us that the model for asthma has been established. Now when we looked at IgE levels of blood and bowel, both blood and bronchoalveolar lavage fluid, we showed that in the disease control model, it was the highest, and it was dose dependently reduced by Withania somnifera and comparable with prednisolone. You can see from the bar chart that all the three graphs on the right side are lower than the black histogram. Absolute eosinophil counts were higher in blood and bowel both in the disease control model. and these were again lowered by Withania somnifera as well as dexamethasone. Interleukin 4 is the Th2 specific cytokine, which is again a biomarker of allergic inflammation. You can see that the IL-4 levels were higher in both blood and bile in the disease control group and reduced by Withania somnifera dose dependently, and prednisolone produced similar results. TNF-alpha, I already showed you, pro-inflammatory cytokine levels were high in both blood and bile, and they were reduced by both Withania somnifera, both doses, and prednisolone. HDSE is a molecular marker of inflammation. So HDSE, inflammatory gene transcription, is regulated by HDSE. So HDSE levels were lowered so HDSE, were lowered in the disease control model. So inflammatory gene transcription was suppressed, as a result of which there was greater degree of inflammation, which was reduced reverted by Withania somnifera in a dose-dependent manner, and prednisolone also did the same thing. Oxidative stress is another biomarker, pathological marker of asthma, and oxidative stress levels were higher in the disease control group, as you can see, from 3.64 to 11.13, and they were lowered dose dependently by Withania somnifera, 2 doses, comparable to that of prednisolone. So we summarize the data. We can see that IgE, TNF-alpha, IL-4, and IL-10, which is the anti-inflammatory cytokines, all these levels were elevated consistently with the disease control model, which were lowered by Withania somnifera, both doses, which was comparable with dexamethasone. Then we looked at airway remodeling. Airway remodeling is a very characteristic feature of asthma, which happens when asthma goes on for a long time because of poor treatment or drug resistance. So as a result of which structural changes occur in the bronchial musculature, as a result of which airflow restriction tends to become permanent, irreversible, and the chances of mortality and morbidity increases. So one of the modes of treating asthma these days is to prevent the remodeling. So remodeling happens because of several biochemical markers, and interleukin 13 is one, which is a fibrotic, pro-fibrotic. All this happens because of fibrosis. So pro-fibrotic cytokine, you can see that the IL-13 levels were higher in the disease control group, that is the OVA group, as compared to the normal controls, NC, and they were reduced by Withania somnifera and dexamethasone. Interestingly, when a subthreshold dose of dexamethasone was combined with a subthreshold dose of Withania somnifera, the effects were complementary, suggesting that Withania somnifera could act as a complementary medication to standard steroid therapy in bronchial asthma. TGF-beta is another pro-fibrotic mediator. So levels of TGF-beta were elevated in fibrosis. So as you can see over here in the airway remodeling, the experimental model for airway remodeling, the TGF-beta levels were raised in both blood and bile fluid, lung and bile fluid, and Withania somnifera reduced both these levels of TGF-beta in both these lung and bile fluid in a manner which is comparable with dexamethasone. And combination of low dose of dexamethasone and Withania somnifera also produce similar lowering effects of TGF-beta. Hydroxyproline is a biomarker of collagen synthesis, and collagen is the prerequisite for airway remodeling. So more of collagen formation would facilitate remodeling. So you can see that hydroxyproline levels were higher in lung tissue as well as bile fluid, and as compared to controls. And these raised hydroxyproline levels were lowered by Withania somnifera and dexamethasone. Also, combination of dexamethasone and Withania somnifera synergistically lowered the levels of hydroxyproline in both lung and bile fluids. Periostin is a new biomarker which we studied for the first time. It is a biomarker for a variety of conditions, and lung fibrosis is one of the areas where periostin is being used increasingly in the clinic. So periostin levels were raised in both lung tissue as well as bowel fluid in the disease control group, that is the airway remodeling experimental group. And these periostin levels were lowered consistently with both Withania somnifera and dexamethasone, as well as a combination of dexamethasone and Withania somnifera in subtherapeutic dose. Then oxidative stress, 8-hydroxy-DG, is a biomarker of oxidative DNA damage. So such chronic inflammation is usually associated with DNA damage as a result of oxidative stress, prolonged oxidative stress, and lung DNA damage was increased in the disease control group. 8-hydroxy levels were raised in both lung and bowel fluid and were lowered by, so showing that oxidative stress induced DNA damage could have contributed to the remodeling, which was lowered by Withania somnifera. This is the histopathological picture. You can see that normal lung in the first one, and then you can see the disease control where the remodeling has set in. You can see the musculature, how constricted it has become, and then treatment with Withania somnifera, and then Withania somnifera and dexamethasone, and then combination of dexamethasone and Withania somnifera. So you can see that Withania somnifera and its combination with dexamethasone has clearly reduced the airway remodeling seen in bronchial asthma. Then the other aspect of bronchial asthma, so this is as far as therapeutic is concerned, a prophylactic, that is prevention of an attack. So prevention of an attack is best, attack is usually triggered by release of biological mediators like histamine and leukotrienes from mast cells. So one of the modes of studying prophylactic agents is to study their effect on mast cells. So mast cells degranulate, and release these mediators which cause the asthmatic attack. So if we are able to prevent the degranulation of mast cells, then we can protect the animal or protect the humans from asthmatic attack. So in the animal model in mice, we showed that degranulation of mast cells were much reduced with Withania somnifera at 200 and 400 milligram, comparable with dexamethasone. So degranulated mast cells were fewer with Withania somnifera. Similarly, such anaphylactic mortality was also lowered, which was 75% in the disease control, was lowered to 50 and 37.5% in Withania somnifera, and was lowest with dexamethasone, just 12.5%. So the anti-anaphylactic property of Withania somnifera is shown over here. To summarize the data, you can see that the aqueous extract of Withania somnifera reversed airflow restriction, showed anti-inflammatory effects and immunomodulatory effects in the OVA model of asthma. Withania somnifera lowered IgE levels, eosinophil counts, TNF-alpha, IL-6, HDAC2 levels in both blood and bowel fluid samples in experimental groups. And these results were comparable with the comparator drug, that is dexamethasone. So this suggests the value of Withania somnifera in allergic asthma. In airway remodeling studies, Withania somnifera treatment attenuated airway remodeling structurally, as well as the biochemical markers of fibrosis and collagen formation like TGF-beta, IL-13, hydroxyproline, and oxidative stress, that is DNA damage. Mast cell degranulation and mortality following ovalbumin challenge in mice was reduced by Withania somnifera, suggestive of its prophylactic value in preventing asthma attacks. The suggested cellular and molecular mechanisms of action of Withania somnifera validate the traditional medicinal effects and strongly suggestive that Withania somnifera can be repurposed for the use in bronchial asthma. However, this is just preclinical data, and randomized clinical trials will further substantiate these preclinical findings and promote integration of traditional and modern medicine. Medicinal plants like Withania somnifera, which has been used for many years in traditional systems of medicine, could have a sustainable impact on health and disease and modern therapeutics in respiratory diseases like asthma. I would like to acknowledge the ICMR, the Ministry of Ayush, Department of Biotechnology for supporting the research, Natural Remedies Bangalore for the generous supply of the standardized extract of Bithania somnifera, the Hamdard Institute of Medical Sciences and Research, PP Chest Institute for providing the infrastructural facilities for this research, and some of my students and colleagues who collaborated with me, other than my co-authors in this particular study. I thank you for your kind attention.